Hello and welcome to another episode of Momento Malum today. In the previous episode, we covered a paper discussing the pedigree and genetic diversity of a small sample of common blueberry cultivars. Due to the smaller sample size, a portion of the groupings on how they relate to each other, as well as the general relationship between the several cultivars, could not be identified. To acquire a greater in-depth analysis of the genetic diversity and population structure of the domesticated blueberry gene pool, the North Carolina State University would conduct a genetic study on a broader population of blueberry lines. The general knowledge of the nature of the blueberry cultivars is that the blueberry cultivars can be divided into two groups, the northern and southern cultivars. The northern stock primarily consists of high bush blueberry lines with incorporation of low bush blueberry in the further north regions of North America. The southern cultivars consist of high bush blueberry lines with frequent integrations of other species such as rabbit-eye blueberry, low bush blueberry, and half-high blueberry, and experimental hybrids. To obtain a superior picture of the gene pool and relationship between the cultivated blueberries, 150 blueberry lines were analyzed. The method of analysis was the use of SSR marker technology in combination with the R software packages known as Vegan and Ape. These were used to study the genetic diversity and the genetic distance respectively. The population analysis was obtained using the software packages known as Structure 2.3 and Clump 1.12. And here are the results. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below. Interestingly, there seems to be some conflict in terms of the gene pool of the northern varieties. In terms of genetic diversity, the wild blueberries are found to have higher levels of genetic diversity than the southern cultivars, and these have a greater level of genetic diversity than the northern cultivars. The high bush blueberry lines were found to diverge into three populations. These include the southern lines, the northern lines, and the lines derived from the cultivar Weymouth. Southern cultivars were found to have a greater level of genetic diversity than the Weymouth cultivars, and the greatest genetic difference between groups occurred between the Weymouth cultivars and the southern cultivars. Southern cultivars were found to be made by mixing northern, rabbit-eye, and other blueberry species together to increase the gene pool. This paper was written in 2014, much later than the previous paper I have mentioned, so there must have been some time to increase the gene pool by a considerable margin. But that's just my speculation on why the southern population now has greater levels of genetic diversity. Both the Canadian and northern United States cultivars in the northern cultivar group show evidence of low bush blueberry integration through cultivated specimens of the species. And these cultivars show evidence of backcrossing back into the high bush blueberry populations. Researchers at North Carolina conclude that they should recommend crossbreeding southern and Weymouth cultivars together, as well as mixing in more wild specimens to increase disease resistance. And that about covers everything. Thank you for watching.